Chuck, Chuck. I'd like to welcome everyone to the Safeway New Employee Orientation. We appreciate your dedication. In all seriousness, we're here to hear Dr. Andrew present on heme proteins. Dr. Andrew grew up and went to college in England, where he obtained an undergraduate degree in chemistry from the University of Nottingham and a PhD in chemistry from Newcastle University. His interest in metal-containing proteins began through an undergraduate research project on heme proteins. And this prompted him to carry out a PhD research project into lobster blood, which contains blue-colored copper proteins. He completed subsequent postdoctoral research at Oregon Health and Science University, and then did some teaching and research back in England. He joined EOU in 2001, and his teaching interests include biochemistry and inorganic chemistry. And he's highly active in undergraduate research. And since 2004, he's actually received external funding from the National Science and Foundation to support it. Last year, he was awarded a sabbatical, and this presentation describes some of those research activities. And in his spare time, he enjoys community choir and long-distance running. Let's welcome Dr. Andrew.
Just waiting for this mic to kick on. I think we're good. All right. Well, thanks, Jared and Steve, for the kind invitation to present my research today. And as Jared mentioned, I'm full circle. Oh, I lost this. Oh, here we go. Oh, here we go. All right, so I've come full circle because, as Jared mentioned, I got into this line of work as an undergraduate in England, and it was through a project on heme proteins, and this is what I currently research. And for me, the involvement in undergraduate research is something I really enjoy here. I've been very lucky that I've been able to teach classes in my favorite subjects, chemistry, biochemistry and inorganic, carry out research and involve undergraduates, which is really what brought me here to EOU. So I'll talk today about some of my research. And in the spirit of a colloquium, I'm trying to make this understandable to a general audience. I realize not everybody's a chemist, so I'll do my best. So this is just a brief outline. I'm going to introduce what heme proteins are, and then talk about light, because this talk is entitled Shining Light on Heme Proteins. And then consider the nitrogen cycle. The biological nitrogen cycle is something I'm particularly interested in at the moment, and involves these proteins. So that'll be my research I will delve into, and a feature of it is that what I really enjoy is involving undergraduates, but also the many collaborations that are involved in that. So I work with people in back in Portland at OHSU, which is where I used to work, but also in England and Japan and France. I'll talk a little bit about that. Last year, I was awarded a sabbatical, and I was able to travel to some of my collaborators. So I will give you a little taster of what went on that year. Okay, so first off, heme proteins. So what they are, proteins are very large biological molecules, and they're made up of amino acids joined together in a chain, and they're folded into a very particular shape, and the shape is important for what those proteins do. Heme proteins have iron in them, and the iron is in the form of this pigment called heme. So this is heme. Fe is iron. It sits in the middle. And this heme is just part of what makes up that protein. And without the iron, the protein wouldn't do anything. It would be inactive, so it's absolutely essential. The other thing about these proteins is that they are in all organisms. They're essential for life in all types of organisms, whether it's plants, animals, microorganisms. So what is the iron doing? Well, it really does two things. One is, like many metals, it can transfer electrons. And so think electricity. And electron transfer is what a lot of these proteins do. And you might know, if you know anything about biology, cell biology, there is the mitochondrion, which is kind of nicknamed the powerhouse of the cell. So in the mitochondrion are a lot of heme proteins whose job it is to basically conduct electricity, transfer electricity in order to generate power. So that's a lot of what heme proteins do. These ones are typically called cytochromes. Another thing that they do, which you're probably aware of, is that they combine small molecules. So an example would be hemoglobin. So here's somebody donating blood, and the red blood cells contain hemoglobin, which is what's giving rise to this red color. And hemoglobin is a heme protein that binds O2, which is oxygen gas, and it delivers it, it carries it around the cell. Actually, it carries it to the mitochondrion, where there are other heme proteins waiting to transfer electrons. So heme proteins are really important. They're also involved in binding other gases. So here we have carbon monoxide, and this is called nitric oxide. Now, if we get too much of these gases, it's poisonous because it stops oxygen binding. But we have, that's hemoglobin. But we have other proteins in our bodies that, for instance, bind nitric oxide. That's what they're designed to do. They don't bind oxygen. Nitric oxide is a fascinating molecule. It was voted Molecule of the Year by Science magazine in 1992. A surprise because it's very toxic, but it was found that it's a key signaling molecule in our body. It controls our blood pressure. It's involved in sending signals in our brain, our nervous system. It interacts with a heme protein. That's how it works. It binds to an iron, a different heme protein from hemoglobin. Some of these other species contain nitrogen, including the NO here. These are found in the biological nitrogen cycle, which I'll talk about, and that has huge implications for life on Earth. And so we'll get there in a few minutes. So what really kind of attracted me to these proteins in the first place is that they're brightly colored, and that's unusual for a protein because most proteins, they're colorless. There's nothing to see. So heme proteins are different shades of red, usually. There are green heme proteins as well. But here is hemoglobin in different forms. So this is somebody donating blood again. This hemoglobin looks quite dark red, purple. It doesn't have much oxygen left. And so really, it's mostly hemoglobin without oxygen at this stage. If it's freshly oxygenated arterial blood, it becomes much brighter red, different shade of red. Here's a blood stain from it's got fresh hemoglobin in. So this has got oxygen bound to the iron. And you might know over time, if you see a blood stain, it changes to a brown color over time. You might have seen that. So this is this case. That's because the iron has lost an electron. And we represent that with these Roman numerals. So iron 3 is one more than iron 2. It means it's lost a negative electron. That changes the color. This is cherry red hemoglobin. This is something you would worry about because this would mean that you've got carbon monoxide bound to your hemoglobin. It changes the color. So the color of heme proteins comes from the heme, this pigment. It absorbs visible light. If you remove the heme from a protein, it would have no color. If you remove the iron from the protein, it's still colored because it's all the other parts of the heme that give the color. But what the iron does is it reacts with molecules, and it changes the color. So heme reactions change the color, and we can see that with our eyes, but we can also use that to monitor the reactivity, and that's what we do in the lab. And we use a technique called UV visible absorption spectroscopy. It's really just measuring the color of heme proteins. So here's our machine here. It's called a spectrophotometer, and we have them at EOU. There's a typical instrument for chemistry students to use. And here's the heme. If something happens at the iron, it changes the color. So what we have are so-called spectra. So when we're talking about absorbing light, here's the rainbow spectrum of white light if you split it into its colors. So everything from red through to yellow-green through to violet, the different colors have a different wavelength of light. And so red has a longer wavelength. You can see this number here, 700. Whoop, sorry. And then violet has a shorter wavelength here, 400. And we can measure using this instrument exactly which wavelengths of light are absorbed, how much absorbance, and we get a so-called spectrum. Each spectrum is specific to each form of the protein. So here we've got iron with one more electron than the red spectrum here. So this is just an electron transfer. It changes the color of the heme. It's like a fresh blood stain versus an old blood stain, and we can detect that. So this is a type of spectroscopy. So this is part of shining light on heme proteins because we can measure the reactivity and we can monitor that in real time and see what's going on at the iron, which is where the chemistry is taking place in these proteins. Okay. So now the nitrogen cycle. So getting towards what we're interested in specifically with these proteins. So the nitrogen cycle is incredibly important for life on Earth. And so what we mean by nitrogen cycle is depicted here in this image is interconverting nitrogen in the biosphere. So nitrogen is a very abundant element, and it's essential for life on Earth. So it's a primary nutrient. It's used to make proteins. It's used to make our DNA. It's used to make chlorophyll in plants. We need nitrogen for that. Nitrogen is very abundant. I think it's the fourth most abundant element on Earth, but most of it is inaccessible to organisms because it's in this form, N2, which is nitrogen gas. That's most of what our atmosphere is. So to make it available, it has to be converted to this molecule, which is ammonia, NH3. And what does that in the biosphere is the nitrogen cycle. It is a series of microbial reactions that will convert different forms of inorganic nitrogen. So nitrogen to ammonia, that process is called nitrogen fixation, and it's catalyzed by microbes. There are other processes which convert to other species. So ammonia, we've got this pathway here, gets converted back to, this is a nitrate. This is the nitrification pathway. And so you can see that not all the fixed nitrogen stays as ammonia. Some of it's taken up by plants, but others are, more of it is converted to other species by these microbes. And then there's denitrification, which sends the nitrogen back into the atmosphere through this molecule here. Nitric oxide, again, the molecule of the year, the molecule that's important for human health, is also part of the nitrogen cycle. So every single step in this nitrogen cycle requires a protein, a metalloprotein. Some of them are iron-containing, some of them are heme proteins, some contain other metals. So it's a feast for somebody like me who's interested in bioinorganic chemistry, because the chemistry is entirely metal protein based. So the current concerns with the nitrogen cycle stem from the production of fertilizers. So ammonia-based fertilizers produced industrially by the Haber-Bosch process. It's very energy intensive, which is a problem because it generates a lot of CO2. The other thing is that the increased amounts of ammonia, it's very inefficient. It's estimated that only about a third actually gets put into plants. The rest of it gets converted by the microbes, which love the extra nitrogen because that's what they feed on for energy and growth. And that then goes to produce these other products. So most of the nitrogen in ammonia fertilizer never makes it to the plants. It goes on to make these species here, nitrate, nitrite. Those species can leach from soil a lot more easily, and so it ends up in water, and it can lead to these dead zones, eutrophication, toxic algal blooms, and so forth. So it's very costly for the farmers that have to buy the fertilizer, and it runs off. It causes environmental damage in the water. The other thing it does is it leads to more of these species in the atmosphere. N2O is nitrous oxide. It's also laughing gas, if you know what that is. It's a very potent greenhouse gas. It's 300 times more potent than CO2. And so increased use of fertilizer has increased the concentration of nitrous oxide in the atmosphere. So there's a lot of concern about how the imbalance of the nitrogen cycle based on fertilizer, and so a lot of research in trying to understand the chemistry of much of which we still don't understand. So one part of it that we've been really interested in is the nitric oxide. We've spent a lot of time looking at this molecule with heme proteins, and this is where our fixed nitrogen can first leave the soil and become and leave it to the gas into the atmosphere. So it's an important molecule for that reason. It also sits at the crossroad of a lot of these different nitrogen cycle pathways, and that's what's shown in this next slide. So back to England. Okay, if you've been to England and you've flown into London, most likely you've had to take public transport. And so one of the train stations, or you might be familiar with if you've read Harry Potter's King's Cross Station. Is it platform 9 3/4? I think it is. So King's Cross is a very busy station, and in part because it sits at the crossroads of a lot of different train lines. So this is the underground, this is the subway, this is a map of the central tube system. Here's King's Cross Station. So each colored line is one of the underground train lines. So those of you who've been to London might recognize some names. So this is the Victoria line, this is the Piccadilly line, the red one is the Central line, so forth. If you want to go from A to B, you have to look at the map and figure out which station you need to go to change trains. You usually have to change and get onto another line. Well, one of the most useful stations is King's Cross because it sits at the junction of, it's got the most connections of any of these train lines. Nitric oxide is a little bit like King's Cross because here's a map of the nitrogen cycle. Okay, oh, I keep doing that, sorry. Here's a map of the nitrogen cycle. So in this case, the colored lines, so they're analogous to the train lines here, are metabolic pathways in the nitrogen cycle. And so here's nitrogen fixation, sorry. The green one is nitrogen going to ammonia, and then nitrification, that's ammonia up to nitrites, that's the purple one, and so forth. Now, instead of stations, we have what are called intermediates. This is molecules that lie along the way towards getting to their products. And so nitric oxide is what we call an intermediate, and it's a central metabolic intermediate. because it sits at the crossroads of a lot of pathways. So you can see it's on the nitrification pathway. Sorry. It's on the denitrification pathway. It's on this NO dismutation pathway. So understanding its chemistry really impacts all the other pathways. So that's important for understanding the nitrogen cycle. It's also toxic, and that's a problem for these bacteria, these microbes, because they can end up poisoning themselves. So how do they prevent that? Well, heme proteins, as well as being involved in these reactions, are also involved with mopping up the NO and stopping it being toxic. So we've been looking at these reactions with NO proteins that are involved in the nitrogen cycle to understand how they work. So the particular protein we looked at last year, which we're still looking at, is called MCCP. It binds nitric oxide. It's believed to be involved in protecting bacteria from the toxicity of that nitric oxide. The particular protein we use is from a methanotrophic bacteria, which is a bacteria that grows on methane. And the particular strain that we study is Methococcus capsulatus Bath. That was first isolated from the Roman baths in Bath, England, which is right here. And the interesting thing about Bath is it's a location of UK's only hot spring. There's only one hot spring in the UK. It's in Bath. The Romans developed it when they were there, and it's a very cool place to visit. This is the bath, the Roman bath. You're not allowed to get in it. It's deemed to be too dangerous, I think, in part because of the bacteria that are growing in it, including this species. So the protein was originally isolated from these bacteria, and we're looking at its molecular structure, its reactivity with nitric oxide, but also the other gases, oxygen, carbon monoxide, to see if we can get some more evidence that NO is important. Is it really binding nitric oxide? How quickly does it bind and release? How does it deal with these other gases? Does it bind them or not? So these are some of the questions we set out to answer. And to do that, we cannot do it all ourselves. It's too much work. We don't have the equipment or the resources. So it involves extensive collaborations. And in biochemistry, typically this is what we do. We're trying to relate the molecular structure of a protein to its reactivity. So if it's a nitric oxide binding protein, that's its reactivity. What is it about the structure that makes it do that and, say, not bind oxygen like hemoglobin? Structure, reactivity. And we also use light, and the interaction of light with materials is called spectroscopy. So the absorption of light that I mentioned is a form of spectroscopy that helps us monitor the reactivity. It can also tell us about the structure. So the reactivity we measure at EOU. We have the equipment to do that. Some of the work is done at OHSU. It's a type of spectroscopy. The structure is determined with my collaborators in England at what's called the diamond light source, which is in rural Oxfordshire. I'll talk about that. And then also actually making the proteins and purifying them. These are a couple of my research students, Jenny Vardenegger and Callie Cruson, who spent a summer in England preparing protein. That was done in England too. Okay, so first off, the reactivity part. Okay, it's UV vis absorption. Already mentioned it. If we can measure how the protein absorbs light, we can track the reactivity. What we're really interested in is how fast reactions occur. And we can do this, in theory, with this machine by taking our protein, taking the absorption spectrum, and then reacting it with something and seeing how fast that absorption peak changes to the other peak. There's a problem here, though. Actually, there's a couple of problems. Even though we can do this for some reactions, a lot of reactions in biochemistry just simply too fast. When you mix A and B, it's over with in a split second. We cannot mix that fast and press scan on the machine. Also, reactions with gases are really, really difficult to do, as you might imagine. They're hard to contain, they're hard to control. So we have a way of doing this. So first off, fast reactions. So fast reactions need rapid mixing. And we can maybe mix in a second, 5 seconds if we're lucky. This machine, it's called a stopped flow analyzer, allows us to mix down to 1,000th of a second. So that's milliseconds. And that means we can monitor reactions on that time scale. So we're very fortunate to have this equipment. So this is just a schematic. You've got two reagents, let's say protein and gas, and there's a drive motor that forces them together, mixes very quickly, and then you measure how the light absorption changes, and it could be right down to just on the millisecond time scale. And so there are various components to that equipment. However, we still need to deal with the gas issue, and that involves using a glove box because we don't want oxygen around, because oxygen is going to react with our heme proteins. And even if we're studying the oxygen reaction, we don't want it there until we're ready to react. So we want to keep it oxygen-free and only add the reagent, the reactant, whether it's a gas or whatever, when we want. So this machine is a glove box for obvious reasons. Here's Jeremy Bard, who was a student a few years ago in Stone Sapphire. And then, so you have to use the stop flow through these big clunky gloves. Inside, there's an oxygen-free environment, and that enables us to carry out our experiments. Now, if you were in the market for a glove box, there's one thing that you really need to check before you buy it. Glove box is quite big. The door to the lab is quite narrow. Our lab is on the second floor of Badgley. Badgley elevator is quite small. Too small for a glove box. So the glove box arrived. We were very excited. In a nice wooden crate. It sat on the first floor of Badgley for many weeks while we scratched our heads and wondered how on earth we were going to get it into the lab. So we were really fortunate that facilities came to our help, and they asked around, and they got the help of a local moving company, Mountain West. So we ended up needing one of these sky tracks, and also the window in Badgley to the lab was removed, and they had to build a special ramp to stop sinking into the grass. Oh, sorry. And very, very carefully put the glove box onto the platform of the SkyTrack and put it, and there it is coming through the window. Now, look at the weather. It's like a blue sky. Okay, at this point, I can tell you it was lunchtime. Okay, so everything stopped. The glove box was hanging half in, half out. Over lunchtime, as often happens in the summer, thunderclouds started brewing over the Blue Mountains. And so it was really touch and go after lunch to get it in before the lightning started. So anyway, it was a happy ending. Here's the glove box safe inside the lab, and we use it extensively. It's really a fantastic piece of equipment, and the stop flow. It's really amazing to have this available at an undergraduate institution. Okay, so we got the glove box. So here's some data. I'm not showing much data today, but here is some data, and I want to show it because it was beautiful data that, I don't know if he's in the audience. Yes, he is. Gabe is at the back. So Gabe Streblow and Tyler Debritz worked with me the summer leading into my sabbatical, and this is one of the many experiments they did that worked beautifully. So we took MCCP and reacted it with oxygen we reacted it with all the gases, nitric oxide and carbon monoxide, but I wanted to show this reaction. So here is the chemical reaction. We are taking the protein. It has iron in it, of course, the heme. It's in the so-called +2 oxidation state. It has to be in this form to bind oxygen. All it's doing, the protein, is forming a bond between the iron and oxygen. Here it is. This is the so-called on rate, and it's also releases oxygen. This is reversible. Has to be, if it wants to be a gas carrier. It's like hemoglobin, binds oxygen, releases it. The other thing that can happen, though, is oxygen. If you think what happens to iron a lot in our atmosphere, it tends to rust. That is oxygen doing its thing and oxidizing the iron. It goes from iron 2 to iron 3. Now, if that happens, the protein stops working. It's not able to bind oxygen in this form here, so it wants to be in the iron 2 state. So what we saw when we did the reaction is summarized here. So before the mixing, so before we've reacted oxygen, here's the red spectrum. So it's got a peak at 431 nanometers. That's the wavelength of light. That's the maximum. When we react it with oxygen, and remember in rapid mixing, 1,000th of a second, it forms the blue species here. That's the oxygen species. Now, it's already happened. We don't see it in real time happening. We just see before, and it's fully bound after 1,000th of a second. This is happening so fast, it's within the mixing time. So we can't measure the rate. All we know is it's faster than 1,000th of a second. It's incredibly fast, much faster than hemoglobin in our blood. Incredibly rapid. And then, after it's formed, in about 15 seconds, it oxidizes. So that's the magenta spectrum here. So we could see this very, very clear, very beautifully in Gabe and Tyler's stop flow experiments. So rapid oxygen binding, subsequent oxidation. We know it's not an oxygen carrier because it destroys the heme, or it sends it to an inactive form. And we know that oxygen binding is very fast. We also did this reaction with nitric oxide and carbon monoxide. They also bind very fast. They don't oxidize the iron, but they bind fast. So our summary here from the kinetics is that the binding of gases is really, really fast to this protein. Happens faster than we can measure it. We would need another technique, of which they exist, but we don't have them. We know it's not an oxygen carrier because it oxidizes the iron really pretty quickly. And we also measured the off rate. So we measured the rate of gas release. I didn't show that. That's also very rapid as well. Those are features that might be useful for a nitric oxide binding protein, because if its job is to mop up this molecule before it's toxic to the bacteria, it can do it fast. And also the rapid release means that maybe offloads it onto another protein to get rid of the nitric oxide. So this was interesting. We wanted to know how the protein structure is affecting the reactivity. And so that's the next part of the talk, is how the molecular structure of this protein was determined. So this is back to shining light on heme proteins. So we've been talking about visible light, or UV visible. That's the absorbance. There was another technique at OHSU, which I'm not going to talk about today, which also uses light in the UV visible range. But light is just, or visible light is just one small part of what we call the electromagnetic spectrum. And so if we think about the wavelength of light, then if it's a longer wavelength than visible light, we move into the infrared and microwave and radio regions. And if we get to a shorter wavelength, we go into the UV, X-ray, gamma ray. And we can use different parts of this spectrum to shine light on heme proteins, and in particular X-rays. So now we're in this very short wavelength, high energy region. And what that's used for is to determine protein molecular structure, the position of all atoms in a protein. So that's what I'll talk about next. So to date, most protein structures that are known have been determined by this technique. It's called X-ray crystallography. It's quite involved. We don't do it here at EOU, but our collaborators in England are experts at it. And so it involves making a crystal of a protein. That's not an easy task, growing a crystal. In the crystal form, if you can produce a single crystal, you can subject it to X-rays. And as the X-rays pass through the atoms, it's called diffraction, which really means that they spread out and create patterns. And if you analyze those patterns of where the X-rays spread out and overlap, it's called a diffraction pattern. You can predict where the atoms were in the crystal. And the X-rays are diffracted by the electrons in those atoms, and you create what's called an electron density map. And some more computation, and you finally get to your atomic model, which is telling you where all the positions of the atoms are in space. The first crystal structure of a protein was back in 1959. That was a heme protein. Myoglobin is the oxygen carrier in our muscles. And since then, the number of protein structures has really taken off. And in fact, there's the so-called Protein Data Bank is a public online repository of all the protein structures that are known. There's currently about 200,000. Since about 2000, most crystal structures have been determined by what are called synchrotrons. And synchrotron is just a way of getting very powerful, highly focused X-rays. So that's what I'm going to talk about next. So part of my sabbatical involved visits to the diamond light source. Now, the diamond light source is in rural Oxfordshire, England. So here's a picture of it. It's a very nice location. It's about 40 miles from Oxford. It is a so-called synchrotron, and it is both a national and an international facility for material science. It's about one of 50 worldwide that are used for that role. So synchrotrons are particle accelerators. So they take charged particles, so in this case electrons, and they move them around at very, very high speeds, close to the speed of light. And because of the circular shape, as the electrons change direction, they change direction because of powerful magnets. When they change direction, they give off energy as intense light, including X-rays. And those are what is used in all the analysis. That's what are used for the crystal structures in our case. So inside the synchrotron, the first thing is it's not really a circle. It's a 50-sided polygon. In fact, I didn't realize it was called this is a new one for me, pentacontagon. Is that right, Steve? So 50-sided. So it's really sections of linear, and then there's a bend, there's a corner, and each time it changes direction, it requires a magnet. They're called insertion devices, and it wiggles the electron, and it gives off intense light. And they're tunable, so you can get X-rays, but you can also get longer wavelengths, infrared. And this is just perfect for X-ray crystallography for proteins. Crystallography of proteins is just part of what this diamond is dedicated to. Most of it is for other applications, but we had about 3 or 4 beamlines that were X-ray crystallography of proteins. So beamlines are where the light emerges, and so there's 3 parts. There's an optics hutch, that's where the X-rays are focused. Then there's an experimental lab, that's where the protein crystal sits. And then because it's too dangerous to be in here because of the high intensity X-rays, there's a separate control cabin, well shielded from the radiation, and that's where people sit. So here, this is a photograph I took inside the control cabin, and this is Is Hans, he's one of the researchers there I work with. He's aligning the crystal in the, getting ready for the X-ray analysis. So he's moving it around until it's just in the right place, and then they're going to collect data. So this is what came out of this, is the structure of the protein. So you get the position of every atom, and this typically is how you look at protein structures, as these so-called ribbon diagrams. They don't show individual atoms, but just so the overall shape of the protein. But you can make out, these are the heme centers. There's actually 2 hemes in this protein. This is an expanded view of the heme, which is where the iron is, where the chemistry takes place. And this sphere here you can make out is the iron. And then this species here is nitric oxide bound to the iron. And these are various amino acids around the heme. And they control the environment of the heme and determine its reactivity. And what makes it different from hemoglobin, for instance, or other heme proteins, is the structure around the heme, the environment. So I'm really trying to wrap this up and cut a long story short. But what we found out from the structure was that the heme is really accessible. By that, what I mean is it's very easy for gases to get in from outside the protein to get onto the iron. In other words, there's a lot of room, and that means that the reactions are fast, and that explains the fast rates that we observed. They were too fast to measure, so that makes sense. Looking at the environment around the heme, it's what we call nonpolar, which means there's no water, and that turns out is not very good for oxygen binding. It means that oxygen binding is very unstable, and so that tallies with what we observed in our reactions, and we can relate that now to the structure of the heme. The other thing we found is that there was an amino acid. This is called phenylalanine, abbreviated Phe. That's the 32nd amino acid in the amino acid sequence here. Now, phenylalanine, for the chemists among you, is an aromatic amino acid. An aromatic carbon has certain electronic properties, and we were able to explain the fast off rates to the presence of this amino acid. Now, to fully explain that, you need to take my course in biochemistry and inorganic chemistry, which maybe you do want to do, but that'll have to be for next year. But suffice to say, we had to go to OHSU and carry out this laser-based resonance Raman spectroscopy to fully understand that. But that's another chapter in the story, maybe for the next year's colloquium. The main result was that this work got published. So here is Gabe and Tyler having a celebratory soft drink, I'm sure. Once this publication, so this was published at the end of 2023, so it's right at the end of my sabbatical, actually involved 4 students from EOU. So Gabe, Tyler is a PhD student at Washington State University. Now Gabe is soon to graduate. He's been accepted into the chemistry PhD program at Purdue University. Sil Vasquez is in medical school, and Zoe Hardy is applying to medical school. So all these students were involved, and so what I've shown you today is just a tiny part of what went into this paper, just to give you a flavor. So really that kind of sums up, here's me outside the diamond light source. It's high security, as you can see. The summary is, hopefully you've got a bit more idea about heme proteins. Hemoglobin is one of them, but there are so many more proteins, and they're involved in different functions. Some of them bind gases, different types of gases. Some of them are involved in catalyzing reactions in our environment, particularly the nitrogen cycle. A major way that we study these proteins is with light, and that's either relying on their color, which is our UV visible absorption, or using X-rays to determine their molecular structure. And there's actually many more that I don't have time to go into, and I didn't talk about what we do at OHSU. It involves collaborations, so we're part of the project, but we also work with the people in England with the synchrotron, with X-ray crystallography, and also I work with my collaborators at OHSU on this resonance Raman technique, which I wasn't able to go into. Collaborators are important. The students, nothing would happen without the students, so everything I do here is undergraduate research, and they're working throughout the year, usually a Friday afternoon or throughout the week, and also through the summer, so I've been able to employ students through research funding, and that's been a real pleasure during my time here. So finally, the acknowledgements. EOU for supporting me, the sabbatical, but also just the fact that we've got an amazing science building with great labs and equipment. My colleagues in chemistry and throughout STMHS have been super supportive. All the students have worked with me since I arrived in 2001. This particular work was Gabe's work and Tyler's, and also Zoe and Sil. The National Science Foundation have been huge, so they funded me ever since 2004. It's through the Biology Directorate, the Molecular and Cellular Biosciences, and they have a program. Oh, I don't know if I've. Oh well. Oh, here we go. Yes. Yeah. So they have a program that is directed to support undergraduate research. And so I have one of their RUI grants, and so that's been a fantastic source of support. And then the collaborators have been Pierre Moen-Lacose, who's a close friend and colleague of mine at OHSU, Hannah Adams at the University of Essex, and Mike Huff is a principal beamline scientist at the Diamond Light Source. And then finally, thank you for being here on such a sunny day, and thank you for your attention, and I'll be happy to answer any questions.
Are there any questions?
Was just part of the MCC PB nonpolar, or is an overall nonpolar molecule?
Just the environment around the heme. So what makes different heme proteins do different things? Because essentially they've all got the same kind of heme. It's just the immediate environment around the iron. And so because different proteins have different amino acids, and those amino acids have different chemical properties. Some of them are nonpolar, some of them are polar, some of them are charged, some of them are neutral. That creates that diversity in chemical environment that then leads to a diversity of functions. So yes, it's the immediate heme environment, which is where the chemistry takes place, which is the nonpolar bit.
And is that unique from like myoglobin and hemoglobin? And
Very unique. So myoglobin and hemoglobin will allow water in there, and since you're a chemist, or you know about chemistry, I can say, well, if hydrogen bonding is very important for oxygen stability. So if you've got a polar environment and hydrogen bond donors, then you stabilize oxygen. If you remove those, or you don't have them there, oxygen won't bind. And so that's one reason that happens.
Thank you.
Mm-hmm.
So when nitric oxide stimulates guanylyl cyclase.
Oh, yes. Yes.
Does it have a heme group?
Does it have a heme group?
Yeah.
Yes, it has a heme group. Yes. So guanylate cyclase is the receptor for, because nitric oxide is a hormone and neurotransmitter in our bodies, in animals. And yes, so hormones bind to receptors. The receptor for that is the heme protein, because iron is fantastic at binding nitric oxide.
And then do we know much about the binding site? So how
Um,
does it
Okay, so
exist for a second or so?
okay, so for guanylate cyclase, so this is our this is what really led to the interest in nitric oxide starting back in the 1990s. Of course, people wanted to know what the protein structure was of the receptor for nitric oxide. And so the problem is that guanylate cyclase, like many proteins, will not crystallize. So when people to try to get the X-ray crystal structure, they couldn't do it. So for a long time, there was no information, very little information. There's a new technique, it's called cryo-EM. It's a type of electron microscopy. That is becoming increasingly important in protein molecular structure determination. Doesn't require protein crystals. The trouble is the resolution is not as good as X-ray. The resolution means can you distinguish one atom from another? And so there has been the cryo-EM structure of guanylate cyclase, but still the exact details, when you're looking at the chemistry at the heme, you want a lot of resolution to see what's going on, and it's not quite there. But yes, heme, iron, guanylate cyclase, yes, that's exactly what's going on, and that's what we have been involved with for, not that protein, but trying to understand how that protein works for many years.
I was just wondering for those kinetics experiments, where less than a microsecond and it's already bound. What temperature were you running at?
Millisecond.
Millisecond.
What temperature? 25 degrees.
So 25
degrees.
Do you have the ability to vary?
We do. We do have the ability, and in fact, we can get information, as you know, from varying the temperature. You can get information, other chemical parameters that tell you about exactly how that reaction's working. But yeah, we can go, we've never done it, but we can go below freezing, in fact, because you put antifreeze in the chiller, which circulates around the stop flow. But so you can go down, I think, to negative 10 degrees and then up to 50 degrees.
Yeah, I mean, I was just wondering, because usually it's a exponential dependence on temperature. Yes. So if you were to cool down, if there's maybe a possibility that you could actually get the reaction to slow down enough to see that binding taking place?
I don't think that's the case, because we do have information about the binding speed, but from a different technique.
Different technique.
So what you can do is you can take the protein with the gas bound, hit it with a laser. The laser causes the gas to come off. Yeah. And then, because laser action happens very, very quickly, you can then measure the binding on down to nanosecond or picosecond, which is even shorter time scales. So we know it happens fast, and it's still too fast for our stop flow.
The system.
Yeah.
Okay.
Still too
and then
fast.
my other related question is, so you have the millisecond and then the 15-second. Did you also collect the spectra in between and see how it transitioned from the bound state to the oxidized state?
Yes, we did do that, and so we were able to get kinetic information and rate constants from that.
Okay, cool.
When you were talking about reactivity and you said that the off rates were also incredibly fast, was that for all of the gases?
That was for all of the gases apart from oxygen, which we weren't able to measure because it doesn't stick around. Although we know what the predicted rate is for oxygen, and that would just be through the roof. So we were able to measure the rates for NO and CO coming off. I didn't talk about that. Relatively fast for off rates, yes, but it's on the minute time scale, something like that. But just fast for an off rate.
So for oxygen, you said you weren't able to actually determine that, but was it able to let go of that oxygen as well as go towards the oxidized
Well,
iron 3 state?
it's an equilibrium mixture that's formed, and because oxygen is going to oxidize the iron, it will eventually oxidize everything there. So yeah, it's the off rate exists, but it's very fast. And we're not able to measure it. All we can see after we mix is the oxygen complex being oxidized. It takes about 15 seconds and it's gone. So if we were mixing this by hand again, the reaction would be over with. So we still need to stop flow even for that reaction.
Awesome, thank you.
Mm-hmm.
You mentioned that the heme proteins are very common among many different organisms on the planet. So, and of course, we have iron.
Yes.
But what is typical what other metals or what other elements will bind in that specific particular heme configuration in other organisms? I'm just curious to know the array of elements that would be part of that complex.
Heme is specific to iron. So
Or
iron.
heme is specific to iron.
Heme means an iron-containing pigment.
But the porphyrin.
The porphyrin
part,
is only for iron. So there are other molecules that look a little bit like the heme. So for instance, chlorophyll in plants, that is somewhat like the heme, only there's magnesium in the middle. But the magnesium's doing something completely different. Heme and all of its reactions, the ability to bind small molecules, which chlorophyll cannot do, the magnesium cannot bind anything. Magnesium cannot transfer electrons. All of that chemistry is not applicable to chlorophyll. So heme, the ability to bind small molecules and transfer electrons, is specific to iron in that case.
So you mentioned you measured reaction rates,
Yes.
and then you, through the structural analysis of the protein, were able to sort of explain why the reaction rates were what
Yes.
they were. Is it actually possible to go in reverse? Like if you know the structure, is there a way to predict what the reaction rates are going to be
Yes.
before you've measured them? And how accurately does that work?
Well, of course, we are always making hypotheses, and we either start from knowing the structure or the reactivity. And we can start from reactivity and say, well, the structure should be something like this based on the reactivity. Or we start from the structure and we can predict the reactivity. But we have to do the experiment. So it's always a case of forming a hypothesis based on what's already known and what we've done before, and then we do the experiment to verify. But yeah, and the other thing I mentioned is that a big part of this is changing the structure. So to have a hypothesis, you say, well, it's because of the nonpolar or the phenylalanine residue. So what we did was change the structure through genetic engineering, molecular biology. So you switch an amino acid, then you determine the structure of that protein and also its reactivity. You have a hypothesis of what should have happened. And so if you think the phenylalanine is there to make the off rates faster, well, you change it to something else, and then you see maybe the rates are not as fast, which is actually what we found. And the structure also ties into that too. So making those structural changes, and constantly you're trying to relate structure to reactivity from both ends.
One more question? Looks like you're free, Dr. Andrew.
Okay, well, thank you.